In this video, we will cover a topic that is global analysis of cell changes in the absence of glycosylation processing proteins. So, what are the proteins that are studied in this project? Is the Calnexin, Calreticulin, and glucosidase 1. So, what is the motivation behind the start of this project? Recently, a paper is published in Science Advance that shows some results that does not have any rationale behind that. So, for example, when the cells are when the spike proteins are produced in these knockout cells, that is in Calnexin knockout and Calreticulin knockout, they can see some viral infectivity from the spike protein, and the viral infectivity from the spike protein that is produced in Calreticulin knockout is very less. Whereas when they produce VLP in the same system, they can see the viral infectivity of the VLPs or luciferin signals of the VLPs is in the Calreticulin knockout is even higher than the wild type, whereas there is uh, almost negligible VLPs production, luciferase signals in the Calnexin knockout. However, at that point of time, we don't have any answer to explain these results. So this is one of the motivation to understand the global changes that happens uh, within the system or within the cell lines that does not have Calnexin and Calreticulin. Another motivation is this recombinant glycoprotein production platform. So how this process happens is that we have one target gene, and then we make the plasmid and then scale up the plasmid and then use any of the cell lines to produce our recombinant proteins, glycoprotein. However, the problem in this different types of cell system is that they are not able to produce post-translation modification similar to human beings. Because of this, it leads to immunogenic reaction whenever some kind of immunogenic reaction whenever this recombinant glycoprotein produced from different cell lines are used by the humans. So right now there is a push to use the cell lines that is derived from a human for this recombinant glycoprotein production. However, the problem with this is that the human cell lines is that they are not industrial friendly. Uh, it means, for example, these human cell lines, the yield from the human cell lines is not so good, and there is still a heterogeneity of this post translation modification that are present in the human cell lines is still exist. So all these things need to be addressed in order to use cell lines that is derived from human for this recombinant glycoprotein production. So this is the study of this post translation modification because of this knockout glycosylation related processing proteins might help to modify these human cell lines that is suitable for recombinant glycoprotein production. What is the let us start with the background of this Calnexin and Calreticulin cycle. So what happens? It begins in the cytosol where this dolichol precursor formation occurs. This half portion of this oligochal precursor forms in the cytosol, and the remaining addition of the this glucosylated monosaccharide happens within the endoplasmic reticulum. After that, this N-glycan precursor will bind to the newly co-translating protein in the endoplasmic reticulum. Then this glucosidase 1 will trim off one of this uh, glucose monosaccharide, and this GANAB or glucosidase 2 will trim off this second glucose monosaccharide, and this monoglucosylated glycan will then be able to interact with Calnexin and Calreticulin, and this Calnexin Calreticulin helps the protein to acquire their native structure by providing them isolated environment, and also allow the other proteins that is not shown here, uh, such as ERP57, to help uh, this newly translated protein to acquire their native structure. And if the proteins successfully acquire the native structure, which is detected, which is checked by this UGGT1, and this correctly folded proteins will then be proceed into the Golgi apparatus through ERIC for further post-translation modification, or and simultaneously for O-glycosylation. And if the proteins does not acquire the native structure, then they will move towards proteasomal degradation through this proteins, and this process happens via trimming of this mannose, as we can see here. So what is the plan that I have used to study these global changes in the cells is that first I did the proteomics analysis, and from the proteomics analysis I made some hypotheses that I used ER stress assay to confirm those hypotheses. Simultaneously, as we know that Calnexin Calreticulin cycle involves in the glycosylation, so disruption in or deletion of those glycosylation processing protein will or should impact glycomics profile. And simultaneously we also check the cell cycle and apoptosis as one of the additional parameter to check the global analysis, global impact of these chaperones on the cells. So first we did the Crispr knockout of the in the hex 293 T cells, and we validated this CRISPR knockout using Western blot, as we can see that this is the Calnexin knockout, this one is for Calreticulin knockout, this one is for MOX knockout, and this one is for dual knockout. This in dual knockout I have knocked out Calnexin and Calreticulin. After that we further verified this knockout using NGS sequencing, and these are the results for NGS sequencing. So we start with the proteomics analysis. So this is the PCA plot of the proteomics analysis. In this PCA plot, this PCA plot represents top 50 variable protein, and we can see that each biological replicates or each knockouts are different from each other and also from the wild type. This indicates that there is some uh, difference in each of the samples, or each knockouts are not behaving in a similar manner. Later on we did check uh, later on we checked some of the proteins. Later on we checked the highly upregulated and downregulated proteins within the proteomics to get an insight about the changes that is going on within those cell lines. So let's start with the Calnexin knockout. So in this we can see that this some of the highly upregulated genes uh, are shown. Some of the highly upregulated genes uh, provided some of the information about have provided some information about what is going on within this Calnexin knockout. For example, this gene that is ATP13A1. This gene is highly upregulated and its function is to prevent cross-contamination of mitochondrial proteins in the endoplasmic reticulum. So this could indicate that in the case of Calnexin knockout, there is a cross-contamination might occurs between mitochondrial and uh, mitochondrial mitochondrial lumen proteins into the endoplasmic reticulum. And simultaneously, another gene that is highly upregulated is the autophagy receptor, and its function is it is usually activated against this viral or bacterial infection. So it is highly upregulated. This could give us two insight. Two things might be happening within the cell Calnexin reticulum cells. First thing is that unfolded proteins might be present within the cytosols, as this autophagy receptor is usually found in the cytosol, and this could also explain why VLP production is low in Calnexin knockout. The third gene that we have checked is GTSC1 gene. Its main function is one of the paper that published is that it prevents the apoptosis by down-regulating p53 function. So what happens in the case when cells are under stress and there is no GTSC1, then this p53 proteins will trigger apoptosis. However, if this GTSC1 is present, then it will inactivate this p53 function. So this is the reason why we do not this is one of the reason why we do not see any apoptosis in the Calnexin knockout. And there are other genes also, and that could be the result of epigenetic changes, because the cells is observing ER, because cells are under unfolded protein response because of the increase in the amount of abnormal proteins within the cells. And when we talk about down-regulated proteins, so in down-regulated proteins also there is WTAP gene is the regulator of antiviral response. So this down-regulation of WTAP usually occurs whenever there is a high production of this IFN1 signal, and this IFN1 is usually high when there is an antiviral response. So this will further substantiate our this will further provide a reasoning for the result that is published uh, in the Science Advance paper, that why VLP productions are low in Calnexin knockout. And the other two genes that have identified is EPHB2 and AR2. These are the glycosylated proteins. So this downregulation of this could relate to downregulation of this to glycoproteins could indicate or could indicates that this Calnexin Calreticulin knockout uh, might affect this post-translation modification of these proteins, and most of these proteins might be degraded via ERAD uh, mechanism. Next we did the Calreticulin knockout. For calreticulin knockout, same this similar observation is observed, that is ATP13E1 is very high. So this indicates that in calreticulin knockout also there is a cross contamination of mitochondrial lumen proteins into the endoplasmic reticulum, and this PPIB gene is also upregulated. Its function is similar to the other chaperones, that it helps the protein in folding and maturation. So this could this could try to compensate the role for the absence of calreticulum, or the absence of this calreticulum in the cell lines. And other genes that are upregulated is related to the PDI family, and these are all involved in providing newly translated proteins, or helping newly translated proteins to acquire the native structure. So this could indicates that there is a unfolded protein response that triggers the upregulation of these genes, that triggers the epigenetic changes that finally triggers the upregulation of these genes. And in case of this calreticulum downregulated genes, uh, there is a downregulation of this general transcription factor. It usually interacts with RNA polymerase 2 and then helps in the translation of the mRNA. So downregulation of this will reduce the translation, will reduce the translation of the proteins that are not essential to deal with this unfolded protein response. And next two genes that are uh, downregulated are the glycosylated proteins. So this further indicates that absence of calreticulum affect the glycosylation of the proteins, or the glycosylated proteins, and most of these proteins might be degrading because they are not able to acquire, and these proteins might be degrading because they are not degrading via ERAT because they are not able to acquire their native structure. Then for MOX, we can see the upregulation of these proteins related to UPR, and which is similar to calreticulum knockout. And next is GTSC1 gene is also upregulated. So this also indicates that it will not trigger, these cells might not undergo any kind of apoptosis. And this HER-C2 is also upregulated. So this indicates that most of the unfolded proteins might be diverting towards ERAT pathway, as the genes that are involved in the protein ubiquitation or degradation is increased. So next is the downregulated proteins in the MOX. So in this, all the three proteins are glycosylated proteins, or all the three highly dysregulated proteins are glycosylated proteins. So this MOX seems to like heavily affected the glycosylation profile, or glycosylated proteins. As we can see, the proteins that are involved in ERAT is also upregulated in this MOX knockout. So last one is the dual knockout. In the dual knockout, we see we have seen some different result. That is, there is upregulation of genes that has this O-glycan site. So in this case, two of these genes that are highly upregulated have O-glycan sites. So these genes might not require this Calnexin and Calreticulin for their processing. That could be one of the reasons why these genes are upregulated in the dual knockout. Next one is this T-SEN15. This is the splicing gene. The upregulation of splicing gene would indicate it does not have any glycosylation site. So upregulation of this splicing gene could indicate that there could be a high translation rate that occurs within this dual knockout. Next one is this Proteomics highly downregulated form genes in the dual knockout. This is the last slide for the proteomics. In this case, we can see that this HMG1 gene is highly downregulated and it is responsible for cellular optimization response. So the downregulation of this could indicate why the cells are not behaving in a logical manner. And the other two genes are related to glycosylation. So this also substantiate the point that due absence of this Calnexin and Calreticulin cycle will affect this glycosylated proteins or secretion of these glycosylated proteins. So what are the main points that we observe in the proteomics is that there could be an unfolded protein response in this knockout, but at different levels, as not all of this knockout shows positive signals for this UPR response or upregulation of genes for this UPR response. The second one is we have seen many glycosylated related proteins are downregulated. This glycosylation profile, glycosylation or post-translation modification might also get dysregulated. The third one is that there is a apoptosis suppression genes observed in some knockouts, that is MOX knockout, Calnexin knockout. So we have used based on these three major findings, we have proceeded we did functional studies in order to check these. We did some functional studies to check each of these hypotheses. So let us start with the UPR response. So this is the basic overview of the UPR response. In this case, whenever the cell experiences a UPR response, it triggers three major pathways. First one is the ATF6 pathway, then IRE1. In the ATF6, what happens is that it is usually present at the ER lumen. So in case of UPRs, this ATF6 will move into the Golgi and there is a proteolytic cleavage occurs, and one of the site of this ATF6 will go go and binds to the nucleus and upregulate this XBP1 or ER chaperones to control this unfolded protein response. The next one is the PERK pathway. In this case, what happens is that dimerization of this PERK leads to the phosphorylation of this EIF2 alpha that will trigger ATF4 to go to upregulate, to go into the nucleus and upregulate the autophagy or ERAD related genes so that they can take care of unfolded proteins via ERAD pathway. Next one is the IRE1 pathway. So dimerization of this IRE1 leads to this XBP1 intron removal, and that will trigger this XBP1 to go into the nucleus and upregulate the proteins that are involved in ERAD or ER chaperones and lipid synthesis. So, and when we check the literature, we are able to get two fluorescence reporter for this. So we check this UPR response with these two fluorescence reporter. These are the two fluorescence reporter that we used. In case of normal processing, only this ORF1 and ORF2 get translated, whereas whenever there is a ER stress, the ORF3 get translated, and we will get a red signal whenever there is a ER stress. In case of IRE1, what happens in the normal case, the plasmid will end here because of the presence of the intron. However, whenever there is a ER stress, this intron get removed and there is a shift of base pair occurs that leads to the translation of this whole plasmid till here, and during this green signal because of this M neon green will also get translated. So first we did the experiment with on wild type cells and this is the experimental timeline that I have used, and for this ER stress or I have used this thepsigargin. It will disrupt the SERCA pathway of this ER that leads to the ER stress generation and trigger UPR response. So these are the InQ side images, and we can see that for this PERC reporter the signals are very bright, whereas for this IRE1 reporter the signals are not that bright. And when we quantified this images through the InQsight software, we can see that for this PERC reporter there is a good difference between the signals are quite bright and there is a difference between negative control and the positive control, whereas for this IRE1 reporter there is a difference between positive and negative control. However, the difference is very low. So we decided to move with this PERC reporter for our knockout experiments, and this is the knockout experiments result, and this is the timeline for knockout experiments. We made a stable cell lines before proceeding for this fluorescence measurement and even did the pyromycin selection. So these are the results on QSight images, results of the different knockout, and this is the quantification of these images, and we can see that Calreticulin has the highest ER stress compared to the others, and dual knockout does not have any ER stress, and this high ER stress in Calreticulin is related to high ER stress is related to the proteomics result. And, we also confirm this result using this flow cytometry assay. In this we can see that Calreticulin has the higher ER stress compared to the other knockout. Later on we check using a Western blot. We check by identifying this. Later on we also check CHOP protein, that is a signature protein which is translated during ER stress, and we can see that the results are in aligned with the in site results that for calreticulin the ER stress is high compared to the other knockout, and for dual knockout there is almost no signal. So these results are somewhat in line with the Proteomics results, uh, where we see in calreticulin knockout where we see the upregulation of other chaperones, whereas the upregulation of other chaperones is not present in other knockout. Next we did the next we did the uh, next we check whether there is a glycosylation related. Next we check the second hypothesis, that is glycosylated related genes are dysregulated. For this we did these two experiments. We did not yet completed this experiment. We did first two experiment, that is cell surface glycan study and whole cell glycomics. The cell surface glycan study, we can see we did the experiment with 23 different lectins and measure the signals using flow cytometers. We can see that there is a significant variation observed in 4 lectins, that and all these four lectins are all these significant changes are found in calreticulin knockout, whereas in all other knockout there is no significant changes has been observed. So this further substantiates the point that calreticulin knockout there is a high there could be some kind of quality control loss happens in the endoplasmic reticulum. However, in other cell surface, for other cell surface glycans, we did not see any significant changes. Later on, we did the whole cell glycomics, and this provides us more insight about the glycosylation or changes in the post-glycosylation or post-translation modification. So in this, we can see that for this dual knockout, there are 23 glycans that are unique to the dual knockout that are not present in any other cell lines. So this indicates that most of the glycans or most of these glycoproteins with these unique glycans might be present within the cells and not on the cell surface. And similarly for these MOX and Calnexin, we can see around comparable glycoproteins, that is 11 and 10 unique glycans are present within the cells, whereas for calreticulin we can only see we can only see 3 unique glycans. So this glycomics analysis provide a better picture that in calreticulin knockout there is less quality control degradation, whereas in other knockouts there could be more quality control degradation within the endoplasmic reticulum. And this figure let us start with this Calnexin knockout. In Calnexin knockout, the changes could be because of this decrease in this hybrid structure, the increase in the complex structure, whereas for calreticulin knockout, similar case could be possible. So for Calnexin, calreticulin behaves in a similar way. And for this MOX knockout, the changes could be The changes could be due to this. Changes could be due to the slight decrease in the complex structure, and this increase in the biantenary structure. And in dual knockout, these changes are also because of this hybrid and complex structure. So most of these changes are because of the change in the hybrid and complex structure. However, in box knockout, these changes are mainly due to this increase in the amount of biantenary structures. And finally, we did also check this apoptosis. Finally, we checked the third hypothesis of apoptosis suppression using two different experiments. First one is the apoptosis assay and the second assay. So, the apoptosis assay, we did not see any significant apoptosis compared to the wild type. So except for this MOX, or you can consider that all of them are behave similarly. So one of the reasons could be that there is a for this Calnexin and MOX, there is upregulation of this GTSE gene that could define why there is no apoptosis. And for dual knockout, we do not see any UPR response. So that could indicate why there is no apoptosis. And for Calreticulin knockout, we see the high ER stress, and the ER stress will work in either of the two way, whether if the ER stress is beyond a threshold level, then it will try to kill the cell via apoptosis, or it will try to restore the cells to its normal state. And we can see the upregulation of the other chaperones and UPR, ER, or this proteins related to PDI family indicates that cells are trying to restore the normal condition of this ER stress. So this is the reason why we do not see any kind of apoptosis. Next, we check the cell cycle assay also. Cell cycle assay also you do not see any significant changes except for this slight changes in the Calreticulin knockout in the G2 phase. And at this moment, this figure is for the cell cycle assay, not for the apoptosis. Apoptosis figures are not shown here. So in the cell cycle, we can see that all the cells behave similarly. All the cells behave similarly, except for this slight decrease in the G2 phase in Calreticulin knockout. And this could be because of the high upregulation of GTSE1 gene that usually works to or usually arrest the cells in G2 phase. Next is the conclusion. So the conclusion phase. What would have happened in the Calnexin knockout WTAP gene downregulation and NDP52 gene upregulation could be the reason why we do not see AVLP production in the Calnexin knockout. For this Calreticulin knockout, we have seen the high ER stress that could relate. Uh, One of the rationale could be that calreticulum has broad specificity and to some extent compensate the role of calnexin. That's why when the calreticulum is not there, we are able to see high ER stress compared to the other knockouts. For this MOX knockout, for this MOX knockout, we have seen the downregulation of glycoprotein genes and even the upregulation of ubiquitination genes. This could indicate that in MOX knockout there is a most of the unfolded proteins are bypassed via ERAD or move into the ERAD pathway. That's why there is a less UPR response, and also since the MOX knockout is the MOX knockout cells, we do not have this monoglycosylated structure, so proteins will not acquire enough proteins will have less residence time in the endoplasmic reticulum. And in case of this dual knockout, we have seen that most of the unique glycans are observed in the intracellular part. This indicates that there is also a loss of quality control in the endoplasmic reticulum, and this is in line with the previous literatures that also reported the same thing. And we have extended that study in a way that most of this glycoprotein that pass or that evade this quality control of the endoplasmic reticulum move into the cytosols or other compartment within the cells. So that's all for this video. Thank you for your time.